everybody. My name is Marija Cvetanovic, and I am Associate Professor at the University of Minnesota at the Department of Neuroscience. I would like to thank the organizer for giving me this opportunity to share our work about selective vulnerability in neurodegeneration with all of you. So what do I mean by selective vulnerability in neurodegeneration? So if we look at most common diseases, neurodegenerative diseases, such as Alzheimer or Huntington disease or Parkinson disease, and if you look at the patients at different stages of progression of disease, you will see a specific patterns in the brain in which specific brain regions and specific neurons within those brain regions are affected. For example, in this slide, we can see that in Alzheimer's disease, here labeled like AD, pyramidal neurons in entorhinal cortex and hippocampus are primarily affected. Similar in Huntington disease, despite the mutant protein being expressed throughout the brain, it is the medium spiny neurons, and particularly D2 expressing, D2 receptor expressing medium spiny neurons in the striatum that are particularly vulnerable in disease. That means they show most pathology and then usually are the earliest pathology and they're the first neurons to die. Now, one thing that you can notice here is there's this brain region at the end, the bottom called cerebellum, and we do not have any specific neurons here. This is not accurate. As you can see in this disease, we do have neuronal diseases such as spinocerebellar ataxia, in which cerebellar Purkinje cells are most vulnerable. So again, here you have cerebellum of the control patients here shown, and cerebellum contains most of the neurons in the human brains. And then you have below the cerebellum in SCA1 patients, which you see a lot of spaces in which cells are missing. And that's even more represented in these images of CRUISE-1, which is the hemispheres of the cerebellum, and then lobule 9, which is a lobule that is in the vermis of the cerebellum, in which you can see that most of these beautiful Purkinje neurons are almost completely gone in this patient. So in these diseases, and there are other spinocerebellar ataxia type 1, these cerebellar Purkinje cells are most vulnerable. So what is SCA1? We'll have a short introduction. This is autosomally dominant neurodegenerative disease, which is caused by the expansion of CAG, trinucleotide repeats, in the gene called ataxin-1. Now, because CAG encodes for glutamine, these end up having polyglutamine repeat expansions in the protein, as shown in this little schematic of the protein. Now, the symptom onset, depending on the number of repeats you have inherited from your parents, can vary, but most commonly it's around 30 years of age, and you show problems with the movement and balance, as shown here with these twin brothers. That also demonstrate that it's autosomally dominantly inherited disease. Now, these symptoms progress. You have also issues with swallowing, slurred speech, and then you progress to not being able to walk, so you're wheelchair bound, have issues with the lungs, you cannot protect them, and then patients usually die within 10 to 20 years of the symptom onset. And as I mentioned, the Purkinje cells are the most vulnerable. And here is the beautiful image of Purkinje cells by Ramon and Cajal. So why does it matter? Why do we even want to explore the selective vulnerability of neurons in the brain? The simplest explanation could be that the cells express mutant protein, only these cells, and that's why they die. But that's not correct. On the top, you can see the Allen Brain Atlas expression of mutant ataxin-1 or ataxin-1 is throughout the brain, so many cells in the brain express it. It's the same with these other diseases here. Mutant Huntington proteins expressed throughout the brain, but only specific neurons are the most vulnerable and show early pathology and loss in these diseases. If we can find why these neurons are particularly vulnerable in disease, then we can devise therapeutic treatments that will then try to make those neurons more resistant. And on the other hand of this is, if we can find what makes the neurons in the other parts of the brain more resistant to disease, we can apply that again to these sensitive neurons and then either delay or completely stop disease progression, which is the final outcome that we all hope we can achieve in these neurodegenerative diseases. Now, one approach we can take is to compare these different neurons. So, for example, I told you that cerebellar Purkinje neurons are particularly vulnerable in SCA1, so I can maybe compare them with striatal neurons that are not that affected in SCA1. However, we do know that there is a large heterogeneity of neurons from these different brain regions, so they come with intrinsically a lot of differences. So it may be difficult to identify the ones that make them specifically vulnerable. The other approach could be to look at the neurons that are more similar. So in that regard, SCA1 is kind of a fortunate thing that there is a difference within the cerebellum in the amount of vulnerability. So on the left-hand image, you see the image of the cerebellum, schematic of cerebellum from Kendall's. And you can see that there is vermis, this middle part is vermis, and then we have hemispheres, and then we have anterior region, and then we have posterior region in the back. So it is the neurons in this posterior region of the cerebellum that are more vulnerable in the SCA1. In fact, the lobule 9 that we took is present in this posterior region. It is the one that I told you is showing almost complete loss of Purkinje cells in cerebellum. So this gives us the opportunity to compare the same cell type, Purkinje cells, in the anterior versus the posterior regions, and ask how are they different? What predisposes these Purkinje neurons in the posterior regions more to disease, and what makes the Purkinje cells in the anterior regions more resistant to disease pathology? The way we do this, the way we study disease and try to ask this causal question is by using mouse models. So the first question is, do these mouse models of disease that exist actually show regional vulnerability that's seen in patient? Because otherwise, if they don't, then we cannot study it in mouse models. Mouse model we decided to use is a knock-in mouse model called the TAXU1, 154Q through 2Q. This notation comes from the fact that 154 glutamine repeats have been knocked into the endogenous mouse locus. So this mutant protein is expressed throughout the brain, which is very similar as in human patients, and it's expressed as one copy, which is also very similar as in human patients. On the right side of the panel, you can see first the knock-in mouse, which has a little hump on the back, which is most likely due to the atrophy and also curvature of the spine being affected, and then you have wild-type mice. So these mice have pretty early pathology. So already at 5 weeks of age, you can see the onset of motor deficits. These mice also have cognitive deficits, and although I did not mention it earlier, cognitive deficits are also present in the patients. And you can see a very visible atrophy of Purkinje cells, which we call here Purkinje cell atrophy, and I'll describe it in more detail in the next slide, already starting from 12 weeks of age. These mice also have premature lethality that's seen in patients, and in this case, in mice, it starts around 28 to 34 weeks of age. So they show most of the phenotypes of human disease, but do they show this regional vulnerability in the cerebellum that we have seen in human patients? So what are the tools that we do to assess the pathology? So for the Purkinje neurons, on the top hand, we have molecular layer thickness. You see these beautiful Purkinje neurons. We stain them with the markers of these cells, and then we see whether they atrophy with disease. It means they kind of retract this process. They become smaller. We also look at the expression of calbinin protein, which is one of the measures that has been shown to track very well with neurodegeneration. We also look at the loss of excitatory synapses. This is something that happens in pretty much all neurodegenerative diseases, that you first lose synapses and then you proceed through atrophy and then loss of the neurons. However, we decided to go further here. We wanted to look how are cells that are surrounding these neurons affected. So we are looking at 2 particular cell types in this study. We are looking at glia, astrocytes, and microglia. For astrocytes, we use glial fibrinolytic protein, GFAP, short, to look whether they show signs of reactive gliosis. Bergman glia is a specific type of astrocytes that's residing right next to the Purkinje cells in this schematic here shown, and they increase their intensity of GFAP staining in SCA1. On the bottom panel, you have microglia density measure as a really easy measure of microglia reactive gliosis. So we count how many microglia do we have in the molecular layer surrounding these Purkinje cells that degenerate. And then we do it throughout the cerebellum. So we do a whole cerebellum image in the vermis. So here is showing you a little bit better. These are anterior lobules, 1 to 5 here presented side, and 6 to 12 we consider to 10 we consider to be posterior lobules. Now, this slide is really busy, so you do not need to pay attention to every single detail. The most point here is we are looking in each of these panels, we are looking at different measure of Purkinje cell atrophy, and then we are looking per lobule. So we're just showing 4 different lobules, lobule 2 to represent anterior lobules. Primary fissure is in between, so that's kind of more posterior, but it's right there at the border, and then lobule 7 and lobule 10 as more posterior lobules. First measure is the measure of atrophy, that molecular layer thickness that I told you, and you can see that it's significantly altered, and to the most extent, the most posterior lobule, lobule 10. In the panel G, you can see, we are here looking at the intense difficult Binet staining, and we see that it starts from, again, first posterior lobule 6, and again is most in the most posterior lobule 10. When we look at the loss of synapses in panel H and I, these are loss of excitatory synapses presented first as a ratio to the molecular layer and then to the ratio how much they're different from the wild-type mice. They are actually, this stage of disease, they are being lost in each of these lobules. However, percentage-wise, this drop is most in the posterior lobules. So this shows us that indeed in this mouse model, we can see the most of Purkinje cell pathology at this disease stage in the posterior lobules, while anterior lobule 2 is relatively unaffected. There is a loss of synapses, but there is not much atrophy seen. As I said, we are interested in astrocytes and microglia, and when we quantify the relative astrogliosis, again measured as increase in GFAP intensity, we can see that only the most posterior lobule that shows the most of the pathology that we could see, we have increase in GFAP intensity. And similar thing is seen with reactive microgliosis. When we quantify the amount of microglia in this, in the lobule per lobule, we can see that only in the lobule 10 in the vermis we can see significant increase, and we have in these are hemisphere lobules, cruise 1 shows some increase. Now from this we can conclude that in this mouse model, we see that Purkinje cells in the posterior cerebellum are more affected, and that this really coincides with selective astro and microreactive gliosis in the posterior cerebellum. We cannot distinguish between whether these reactive microglia and astrocytes are causing Purkinje cell in cerebellum to be more atrophied, or it's vice versa. We can just say that they coincide at this point. But the most important point from this is that we can reproduce what's seen in patients, that most posterior regions are more vulnerable, and therefore we can use these mice to investigate the molecular mechanism and also functional consequences which may underlie this selective vulnerability in SCA1. So first we want to look at the molecular mechanism. So how do we develop the tools to look how are these regions different at the molecular level? So very talented student in the lab, Katie Hamill, developed this very easy and quick dissection of the cerebellum into the anterior and posterior regions for molecular analysis. So she would very reproducibly be able to separate these regions. And then she would extract RNA, we would do RNA sequencing. Now, this would allow us to probe and ask many questions. First question that we wanted to ask is, how are these regions different in wild-type mice? How does these differences may predispose the posterior regions to disease pathology? And then the second question is, how do these regions differently respond to disease pathology in ataxin 1 mice? So for the first question, what are physiological differences in the wild-type mice? We have compared these regions, so anterior versus posterior. So this means cerebellar cortex posterior vermis and cerebellar cortex anterior vermis. And this, on the left, you have volcano plot, which shows us differences in expression of genes. And on the right, panel G, we have KAG pathway analysis. So one of the things that we were really astounded when we did this analysis is that we found a large amount of differentially expressed genes between anterior and posterior vermis. This is intracerebellar region differences. So we did not expect to find around 1,600 genes that are different between regions. You can see here that there is volcano plot. There are many genes that are differentially expressed, and most of them are actually decreased in posterior vermis, meaning that they either increase the anterior vermis and show downregulation in posterior, but there is also the other way around. We have genes that are more expressed in posterior vermis and less in anterior vermis. Some of these genes may predispose posterior vermis to disease, and some of them make anterior vermis more resistant to disease. Similarly, when we look at the KEGG pathway analysis, we see that there is large differences between these regions in the wild-type mice, so this is without any disease conditions. And specifically what we found really to be interesting, that pathways of neurodegeneration, so things that are activated in many neurodegenerative diseases, genes that belong to these pathways are differentially expressed between more vulnerable and more sensitive regions of the cerebellum. Now, this shows us that some of these genes may predispose it, so we then wanted to confirm some of them, and one of them was cholecystokinin. So some of you may know what cholecystokinin is, some don't, so I'm just going to give a really brief introduction. This is a neuropeptide that has been shown to show some neuroprotection. In particular, it has been shown to show neuroprotection in spinocerebellar ataxia 1 and spinocerebellar ataxia 2. So we wanted to confirm this, and we then measured the amount of CCK levels in the anterior versus posterior cerebellum and hemispheres. And what you see here on the right panel, this is qPCR, quantitative real-time PCR, and in the black is shown wild-type mice. You see that we have significantly around 50% more of CCK expressed in the anterior vermis than in the posterior vermis. And then this goes in disease, it decreases in both of these regions, but this decrease is even more in the posterior region because we start from much less. So CCK may be one of the factors that underlie resistance of the anterior vermis. Now, next question we wanted to ask is, do these different regions within the cerebellum respond to disease pathology in the same manner, or are there differences? So again, we were really surprised by the amount of differentially expressed genes. So here in panel A, we have a Venn diagram which shows us how many differentially expressed genes between different cerebellar cortical regions are shared and how many are unique. And it comes that about 1/3 of them are shared. So in the anterior vermis, we have around 1,650 genes that are differentially expressed in disease conditions, and 683 are shared with all three different regions, including hemispheres and posterior region. This also tells us that similar amount of genes is uniquely perturbed in each of these regions. So in anterior cerebellar vermis, we have around 663 that are affected only in that regions and around 503 in the posterior cerebellum. So this is really surprising that within the cerebellum, there is such a large difference in disease, in response to disease condition. What was even more surprising when we did pathway analysis, so I know these are really tiny, so the first row column here shows anterior cerebellar vermis, all the pathways that are affected there, and the second one is posterior. Remember, again, it's posterior cerebellum that has the most pathology in this disease. And what is really easy qualitatively to see here, even though you may not see the small differences, is there is significantly more affected pathways in the posterior vermis that shows more pathology at the level of histological changes such as atrophy. So we see that these molecular changes are really following up with what we see at the level of pathology, and there is a wide variety of changes. So next step is to look at the cell type specificity and how changes in each of these cells that I mentioned to you. So we have Purkinje neurons that are mostly affected. Then we have also Berman glia that I told you are also affected, and we do not know which one causes what. So we wanted to look at specific genes which we know are really important for the function of these cells. So for Purkinje neurons, we know that GRID2, which is glutamate receptor, we know that it is significantly affected only in the posterior region. Now, GRID2 is mutations lead actually to ataxia in mice. In fact, this is the cause of spinocerebellar ataxia 18. So what we have been able to reproduce here, that while it's not at all affected in the anterior vermis, it is significantly downregulated in the posterior vermis and the hemispheres. Then regarding Berman glia, we decided to look at the GLI1. GLI1 is a transcriptional factor which is involved in synaptic signaling. We know that Purkinje cells signaling through sonic hexog into the Bergman glia is really key to their function. So it's really necessary to maintain the Bergman glia function. Bergman glia function is there to support Purkinje cells. So if something goes wrong with them, that is definitely going to influence how Purkinje cells function. So we wanted to see whether this is affected, and this is one of the genes that came up in our RNA sequencing as being decreased only in the posterior vermis. And then indeed, using qRT-PCR, we have been able to see, to validate this, that there is a decrease in GLI1 expression only in the posterior vermis, but not in the anterior vermis Bergman glia, indicating again that there is this cell type specific changes in anterior and posterior vermis that could be responsible to the increase or selective vulnerability of posterior vermis in this mouse model. So our conclusion from this second part is that RNA sequencing can identify unique pathways that are seen in wild-type mice in physiology and also in a disease model of mice. And they are different in susceptible and resistant cerebellar regions. And then the genes that are specific to different cell types, such as GRID2 in Purkinje cells and GLI1 in Bergman glia, are also regionally regulated in physiology and in SCA1 disease, and maybe again underlying this vulnerability in disease. Now, the next big question is, so we have seen changes at the level of pathology. We have seen changes in molecules that are expressed in these cells. Does this lead to any functional changes? In the effect, the disease symptoms that we see are likely seen because Purkinje cells or neurons are dysfunctional. Is there change in functionality in Purkinje cells in the anterior versus Purkinje cells in the posterior region is the key question, because that is really what will influence the behavior. So in order to understand functional implications of this vulnerability in disease, we decided to look at the calcium signaling. Calcium signaling has been proven by many researchers to be a really good measure of neuronal activity. Now we wanted to look at the calcium signaling while mice are freely moving, so they're doing motor behavior for which cerebellum is mostly responsible. To do this, we are using genetically encoded calcium indicator, or GCAM6F in this case, and we are expressing it primarily in Purkinje cells using PCP2 Cre mice, and then this is all done in wild-type or ataxin-1 mice. To look in the freely moving mice, we are using fiber photometry because it allows mice to be recorded. They don't need to be anesthetized while they are freely moving. So on the left, you have the schematics of how fiber photometry normally works. And then, so it is getting the signal from the population of the cells. It's not just Purkinje cell, but population of the cells expressing it. And then on the right, you have image of Purkinje cell, mice with fibers implanted in the lobule 4 and 5, so anterior lobules, and then lobule 6 for the posterior lobules. The reasoning we do 4 and 6 and not 2 and 7 is because of the anatomy of cerebellum precludes you to record from these without damaging the other lobules, and we want to try to avoid a huge cerebellar damage. So here are results. So here is presented the frequency of the calcium signaling in the Purkinje cells in wild-type mice in lobule 4 and then lobule 6. So these are presented in green. Lighter green is lobule 4 and darker green is lobule 6 to kind of present anterior to posterior. And then we have ataxin-1 mice in lobule 4 and lobule 6 presented with the lighter and darker red. And you can see that between wild-type and ataxin-1 mice, SCA1 mice, lobule 4, we do not see a significant change in the frequency of the calcium signaling in Purkinje cells. However, we see a significant decrease in the frequency of calcium signaling in Purkinje cells from lobule 6 in wild-type mice to the lobule 6 in the SCA1 mice. These results indicate to us that posterior lobules have decreased frequency of calcium, so they are the ones that are dysfunctional in disease, correlating with all the previous dysfunction that have been seen at the pathological and molecular level. While anterior lobules, in which we have seen much less pathology, both at the pathological levels and also at the level of molecular changes, are mostly functional. They mostly function as seen in wild-type mice by this measure of Purkinje cell function. So for this, we conclude that this mouse model displays differential Purkinje cell degeneration across the cerebellum with increased vulnerability in the posterior vermis and relative resistant degeneration in the anterior vermis. And then that we can use molecular and functional assay to further probe that. So RNA sequencing also demonstrates that posterior vermis is more significantly impacted. And also fiber photometry, imaging of the calcium activity, shows us that Purkinje cell function is more impacted in the posterior lobules. This gives us a really good system in which we can then probe for causal. We can manipulate certain genes and pathways then to see if we can rescue this selective vulnerability in the posterior vermis and make them as resistant to disease as the anterior vermis Purkinje cell. But the big take-home away message, which are a little bit further away from SCA1, is that this inter- and intra-regional heterogeneity of neurons and glia in a brain is key characteristic of a healthy brain. But also selective regional neuronal vulnerability and gliosis is also a key feature of neurodegenerative diseases, so they go hand in hand. And the molecular differences underlying this heterogeneity of the brain likely contribute also to selective vulnerability in the brain disease and can be the good targets in which we can increase resistance of neurons in disease, or we can increase resistance of glia to disease. And then the larger cellular molecular changes will correlate well with neuronal dysfunction in vulnerable region, indicating that there is a really threshold for dysfunction and or compensatory changes in disease-resistant regions. So I would like to thank you for your attention. I would like also to acknowledge all people in the lab, most importantly, Katie Hamel, who is sitting in the second image by the table. She is a graduate student, soon to graduate from the lab, very talented, and she has performed most of the work here, except Francisco Labrada Monseada is our very talented postdoc who did the fiber photometry recordings. I also want to thank all the people in our lab, with whom we very closely collaborate, and our lab creative catalysts shown on the left, Birdie, Jumbag, Laszlo, Millikan, and Damples. I, of course, want to thank all the funding agency without whom this work would not be possible. And again, I want to thank you for your attention.